Okay, let's continue. So, yeah, like we've been saying, there are a lot of different variants. You have multiple copies of the same thing. It's what we would call redundancy built into the genome so that you have different possible ways to make an immune response. You're basically taking multiple lottery tickets to increase the chances that you win the genetic lottery, essentially. That is the idea. And yeah, they can move around a little bit uh, as well. Okay, so isoforms. Yeah, fine. So different forms of the same protein. Yeah, like different alleles, like an HLA A star 01 versus an A star 02, for example. A sequence motif is going to be the little pattern of amino acids that determines what a particular MHC type can actually bind to. And so for each isoform of each of the types of HLA molecule, you're going to have a slightly different sequence motif pattern. Each one of these can bind a slightly different set of peptides, which gives you a potentially very broad set of immune responses that a person can make. And potentially here with genetic modification, you could start to add some of these extra sequence motifs. You could take some of the most useful ones and put those in addition to whatever types people are getting from their parents. And yeah, potentially have a more resilient human population. This is something that would probably work. But again, yeah, you can't just go around genetically modifying people. There are laws, there's public opinion, there's consent, and a modification like this would be permanent. So, yeah, there's a lot to think about there. But this is something that science may be moving toward, at least as far as I can tell. Okay. MHC restriction says that no matter what peptide you've got in your cell, if your MHC cannot bind to it, you're not going to be able to show it to a T cell. So you could be rampantly infected with something, and if you happen to be born with the wrong MHC, then you're going to be restricted because each MHC can only present certain peptides. Each proteasome and immunoproteasome can only cleave certain peptides. And if you don't happen to be able to load the peptides that you've cleaved, or if there don't happen to be a good set of peptides coming from a particular protein from an infection, then there's going to be no presentation and no adaptive immune response downstream of this. So yeah, this is big. All right. So yeah, Rolf Sinkernagel. Yeah, okay. Who's an old virologist actually, and Peter Doherty, who I have not met, showed that yeah, antigen recognition is MHC restricted. I think Sinkernagel published the paper showing the difference between CD4 and CD8 or something like that. Just really basic stuff a while ago, and really short paper, but extremely important in the long term. All right, so MHC polymorphism, like the different types, is mainly going to be concentrated up in this area. So right in this little red area at the top in the alpha 1 and alpha 2 subunits. So each of the little red dots shows you a place where you're going to have some changes from one molecule to another. And so this looks like class 1 over here, and you got your class 2 right there. And yeah, once again, you're going to have changes in the bottom and changes in these two alpha helices that are going to pinch the peptide. And so this is where we get different versions of these proteins that are able to present different peptides. And once again, if you were to tinker around with molecules like this, theoretically you could figure out ways to present other peptides that human genetics has not allowed us to present. So potentially you could make a version of an MHC class 1 or 2 that's able to present things that people can't otherwise present. That could be a very big deal to the survival of the species, for example. All right, so here are some of those little peptides, and we're showing the different positions. So position 1, 2, 3, 4, 5, 6, 7, and 8, and how they bind, and we're showing that of the ones that are binding to this particular MHC molecule, you're going to have a couple of these anchor residues where you're going to have similar things. So L is leucine, I is isoleucine. They're almost the same. F is phenylalanine, Y is tyrosine, which is phenylalanine with a little hydroxyl group on the end of it. So pretty much the same. And then over here we've got generally hydrophobic amino acids on position 2. And yeah, so this is the KB version. This is the KD version. So we're talking about mice here. And you can see that we've got, the anchor residues are shifted over a little bit, 2, 5, and 9 instead of 2, 5, and 8. And yeah, we have anchor residues there. And so shown in the little yellow box, you can see that you can have bacteria, and you can have viruses, and you can have regular host proteins. And all of these can be chopped up and presented on these particular molecules. They all happen to have little peptides that can be presented on these molecules. So this one down here, LCMV, is one that I've worked on before. It's a mouse virus that can sometimes transfer over to humans. It's actually pretty common in mice, and when it gets into a person, it can be deadly. One of the ways that this gets in is that a person will not know that they're infected. They often wouldn't screen for this at a hospital, and then they do transplants. And what you find out is that each person that gets a transplant then goes on to die from LCMV, because when you get a transplant, you're also on immunosuppressives. And so you shut down the part of the immune system that would ordinarily be dealing with a pesky little virus like this one. So there you go. It's kind of neat. All right, so MHC restriction. The entire principle is that unless you can make a good junction, so you've got to have the right peptide that fits into MHC, and the peptide and the MHC molecule have to be able to bind to the alpha beta T cell receptor, let's say here, or you would have complementary versions binding to other kinds of T cell receptors, like the gamma delta. So one of the MHC versions that is the sort of low, sort of static, low variability form. This is going to present peptides that potentially can't bind to the T cell receptor, and so bam, no binding. And your other versions of MHC, the odd invariant versions, could present a peptide that would totally bind to a T cell receptor, but they themselves may not be able to dock with that T cell receptor. So yeah, if you got the wrong type of MHC, or you've got the wrong kind of peptide in the MHC, then even though you presented a peptide correctly, there's going to be no recognition by that T cell. And so we call this MHC restriction. You got to have the right context, the right MHC, the right T cell receptor, and the right peptide. Yeah, it's all got to be going right in order for us to make what we call an immunological synapse, or synapse, if you like, which is just where the antigen-presenting cell and the T cell are going to come together and actually make contact and, interface and share peptides and information. Okay, so let's carry on. Okay, alloreactive T cells recognizing non-self MHC molecules are very abundant. Yeah, okay, fine. So T cells recognizing MHC molecules that, or rather that are MHCs that are presenting molecules that you don't make in your body normally are going to be fairly abundant, yes. Allogenic is, like having different MHCs, so mixed lymphocyte reaction is going to be a way that you can test, right? For tissue compatibility, you could put T cells from one with the MHC molecules of the other and see if they are perceived as foreign, or if they are, or if they just, if they work together, if they're perceived as self. And so in general, the T cells are not going to attack self antigens, and they are going to attack foreign antigens. Attacking self antigens would be a thing called autoimmunity, and that's something that you really don't want, and that the immune system is going through a lot of steps to try to avoid. So, alloreactivity is recognition of non-self. That's what you want. Positive selection is where you actually negatively select, kill off T cells that recognize antigens presented by the antigens from self proteins. So you don't want your T cells attacking yourself. You want them attacking proteins that are coming from foreign invaders. And so the entire selection process that's taking place there is what's going to determine whether you have a lot of auto-reactive T cells and auto-antibodies floating around in your body, both of which are potentially going to lead to autoimmunity. Yeah. So here we go. This is showing one way that we can make contact between the T cell and the MHC molecule. So this is the complementarity determining region, or the hypervariable region if you like, of the T cell receptor, and you can see it's got a couple amino acids in the top, and you can see that they are making contact with that big helix at the top of the MHC molecule. And so boom, these are the contacts that you have to make in order to actually get these two molecules to link up. All right. And we'll go a little further, and then we will pause this section and finish it out in one more section. So, minor lymphocyte stimulating antigens. This is an old term for the non-MHCs or non-canonical MHCs that are responsible for strong T cell responses. Now these are known as super antigens. There are some antigens that cause just really strong responses. And endogenous retroviruses. These are old virus-like sequences that you can find embedded in your DNA, and they actually outnumber our genes by a considerable amount. But yeah, we have lots of these copies buried in our genomes, and so these are some of the things. Let's see, staphylococcal enterotoxins, fantastic. So these are going to be involved in food poisoning. They're going to be secreted toxins. Staphylococcus is one of the things that can occasionally turn into the flesh-eating bacteria if it gets into a certain layer of your skin, for example. And they secrete all kinds of toxins that can break down your tissue and cause horrendous swelling and stuff like that. So just really strong inflammatory promoting toxins. Yeah. And then toxic shock syndrome is when you get the full body basically in an inflammatory state, and it is usually very serious and quite often fatal. And so one of these enterotoxins is going to be toxic shock syndrome toxin number 1, which does cause toxic shock syndrome. And so like we were saying, toxic shock is when the entire system is reacting and just producing massive amounts of cytokines with your CD4 T cells, and this TSST1 protein from Staphylococcus aureus is going to be one of the bacterial superantigens that just sets off an enormous immune response, mainly CD4 mediated. Okay, so what this is saying here is that lots of T cells can respond to some of these super antigens, like the TSST1. And so the way this is going to happen is that you'll have your antigen presenting cell. You'll be presenting class 2, just like a good little antigen presenting cell. It'll be binding to the alpha beta T cell receptor. But then you'll have your super antigen that'll actually come in between. It'll actually make the junction between the T cell receptor and the MHC class 2. And we're not sure if the protein has evolved to do this in order to kill the host, or if the protein just happens to do this and it's like an accidental thing that happens in the world. I'm not sure that's been adequately described. Shown over here, this is the actual the chain. So here's our peptide, here's our MHC molecule, class 2, so we got our alpha and beta chain, and we're pinched between there. Here's our super antigen kind of docking in between these two things. So even if this T cell wouldn't interact with this peptide, the super antigen is going to make it interact with the MHC molecule, and it's going to set off the same sort of cascade. It's just that it can make every MHC bind to every T cell receptor, and so that's why you get a really big response out of this. Yeah. All right, so that's a good place to break this section, and we'll come back for one last section where we're finally going to at least mention gamma delta T cells. So yeah, let's look forward to that and talk a little about NK cells.